Hey everyone, thanks for joining me today for episode 778 of People Behind the Science. I'm your host, Dr. Marie McNeely, and I'm excited to welcome our guest, Dr. Rachel Bennett, to the show today to talk about life and science. This episode is made possible with support from our sponsor, Innovative Research. Listeners, Innovative Research has provided scientists with quality lab products made in Novi, Michigan for more than 20 years. Their portfolio includes human and animal biologicals, assay and ELISA kits, antibodies, and more. Products can be purchased directly from their website by credit card or purchase order at www.innov-research.com. That's www.innov-research.com. They offer small quantities, bulk purchases, and custom orders. From human whole blood to specialized antibodies, Innovative Research has what you need to succeed in the lab. And today, Rachel is going to tell us some great stories about her lab and her life. So listeners, get ready to meet another one of our outstanding people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am thrilled to be speaking with our guest researcher, Dr. Rachel Bennett. So, Rachel, welcome to the show today. How are you?
Hi, doing great.
Well, we are so excited to have you here with us today. And listeners may know that we actually went to graduate school together at Washington University. So I know a little bit of your story, but we will share some of these details with our listeners today. And I expect I will be learning something new as well. I'd like to start by telling our listeners a little bit about you and how you got to where you are today. So, listeners, Rachel is an assistant professor in neurology at Massachusetts General Hospital and Harvard Medical School. She received her bachelor's degree in molecular biology from Arizona State University and her Ph.D. in neuroscience from Washington University in St. Louis. Afterwards, Rachel conducted postdoctoral research at Harvard Medical School and Massachusetts General Hospital before joining the faculty there. Rachel has been named an outstanding emerging scientist by the BrightFocus Foundation, and she received the new Investigator Award from the Charleston Conferences on Alzheimer's Disease, or CCAD. And to help our listeners get to know you a little bit better, Rachel, can you start by sharing what you like to do outside of science?
Yeah, I grew up on the west coast of the U.S., so I miss the mountains. I currently live in Boston, Massachusetts, and I really miss going out and doing some hiking. So more recently, the last couple of years, I've started tackling, there's this 48 peaks over 4,000 in New Hampshire. And so I've been trying to tick those slowly off the list. I haven't gotten very far, but if I have some free time, I like to just kind of go out into the mountains and do some hiking when I can. And aside from that, we're also next to some really beautiful beaches here in Boston, Massachusetts. Not typically what you think of when you think of beaches, I guess, but I'm just happiest bobbling around like a clam in the ocean. So,
Well, I love it. I think getting outside can be a good way to kind of refresh your mind, take a break from science for sure.
Otherwise, being in the lab kind of looks the same in any season. And we spend a lot of time indoors, spend a lot of time staying at a computer screen or sitting at a bench, and getting outside definitely is a good reset button for me.
Absolutely. So let's talk about what you do in the lab at the bench then next. How do you describe your science to someone who's not in your particular field or not particularly familiar with your research area?
I'm an Alzheimer's disease researcher, and I'm interested in understanding how blood vessels in the brain change in disease and how that might contribute to the loss of neurons. And then otherwise, if you ask my son, he's 11, I spend a lot of time looking at brains, is what I tell him.
I love it. So is your son getting into research as well?
He's kind of as far from me as could be. He really likes history and geography, but he didn't quite get the science bug. So it's kind of interesting. It's the nature versus nurture argument.
Well, it's wonderful to hear a little bit more about what you do in the lab. And we'll go through some of the details as we go through our conversation today. But we love to start our interviews by talking about motivation. I think as a scientist, you go through a lot of challenges, and sometimes every day feels like a challenge. So it can help to have a little piece of motivation or inspiration to keep you going when the times are tough. So Rachel, do you have a favorite quote or a saying or just motivational force that really keeps you inspired?
Well, Marie, I'm not a huge motivational quote person, but something that always comes to mind, and this is a little bit silly, I think of Yoda and I think of Star Wars and do or do not, there is no try. And that kind of comes up every day. It's a funny thing to think about in science because it's pretty much all try in science. There's a lot of doing when you get to it, but for the most part, there's a lot of trying and failing and trying and failing. And when we're thinking about Alzheimer's disease research in particular, ultimately my goal is I do want to contribute to a cure for Alzheimer's disease or work towards that. I think that is a little bit like Luke Skywalker in the swamp trying to raise the X-Wing. It seems a bit impossible at some point. So you kind of have to go in with that mentality of like, yes, I can do this. But on the other hand, there's going to be a lot of failure and just trying and restarting over and over again. So it's a bit aspirational.
Oh, that makes sense. And I think it definitely applies in science. So as a scientist, you get to work with a lot of really cool people over the course of your career. And some of these people can also be sources of inspiration or motivation. So when you look back at your path, Rachel, are there particular people who come to the top of your mind as mentors or role models or people, whether inside science or not, who you looked up to?
Yeah, I've been really fortunate to work with really great folks over the years. So there's a lot of people that I definitely look back on, and they've certainly influenced my career and my career trajectory. Starting at ASU, I had this great teacher. Her name was Jane Mindshine, and she taught this just absolutely phenomenal history of science course. And that really stuck with me. She recommended a lot of reading, so a lot of just different writings and really putting the history of science into context for us and made me think about it in a different way that I think ultimately kind of contributed to me pursuing a career in science. And after that, she also served as a mentor to me. I was pursuing a honors thesis project, and she was the one who recommended that I apply to a travel scholarship to go attend a AAAS conference. So it's a big scientific conference, and it was actually held in Boston that year. And it's not something that I would have done otherwise. So she really just kind of like pushed me over the edge and pushed me to really lean into my interests at the time and apply to graduate schools. So I really have to thank her for that and for kind of getting me to where I am today. Then in grad school, I had a really excellent graduate thesis mentor, David Brody, and he really impressed upon me this idea of performing rigorous science and really thinking about the statistics and thinking about the experimental design. And I have to say, I've kind of carried that with me since that day. And then my current mentor is Bradley Hyman, and he's really kind of a top-notch expert in Alzheimer's disease. And something that I've taken from him is just this idea of just going where the science leads you. And we can get a bit of tunnel vision sometimes with our projects, but he's really been the one to kind of point out, hey, actually, this thing that you just told me is really interesting. Like, tell me a little bit more about that. And he's given me this confidence to kind of go outside of my comfort zone and really pursue the research questions that I find the most interesting. So just kind of as a composite, these three individuals looking back have really served as this template for a role model for me. And that's really looking forward to my career, the people that I'd like to kind of emulate moving forward as well.
Oh, that's wonderful. And I like that you mentioned these three mentors at different stages of your career path. And I think that's something that I've heard from a lot of scientists. You sort of find a person at each different stage and gather a couple of their traits or lessons learned along the way. And that helps you become the scientist that you are today. And I really like this idea that you mentioned of just with Jane Mindshine, understanding the stories behind research, not just perhaps memorizing a collection of facts, which I feel like a lot of undergraduate coursework was. So let's talk about your maybe early days as a scientist, even perhaps before college. What really got you started on this path, Rachel, and turned your eye towards science and perhaps neuroscience?
I think when I graduated from high school, that was about the same time that the first human genome was being completed and published by the Human Genome Project. That was about 2003. So that really kind of set the stage for me and really made a huge impression on where I started my career. I applied to Arizona State University and was accepted into their molecular biology program. And at the time I thought, okay, great. We've got this human genome. It's going to provide this blueprint that's going to answer all of our questions. So I thought we're going to go in, we're going to cure all kinds of diseases using gene therapy, and just really was fascinated by this idea that was emerging at the time. And kind of through the coursework and just some of the practical hands-on research that I was doing at the time, it quickly became apparent to me that molecular biology was not necessarily a good fit for me. It was a lot of pipetting, have invisible liquids from one tube into another. And to me, that just wasn't super gratifying. So I quickly pivoted to this more of a cellular biology aspect. And I just fell in love with kind of using different microscopes and just seeing changes as they were occurring in the brain. And one of my projects that I was doing as a graduate student at Washington University in St. Louis was trying to just visualize axon injury and concussion. So concussion often leaves these invisible lesions on the brain. And I was just really fascinated by this idea of trying to make that injury visible in some way, shape, or form. And through that project, I've learned to do all kinds of things like use an electron microscope, which is a really fancy microscope that shoots a beam of electrons. So these have a wavelength of about 1 nanometer across. And so you can resolve these super, super tiny structures in cells. And I was using that to look at these super tiny axons that were less than 500 nanometers wide in the brains of these injury models that we were using. And it was just fascinating to me. I think I probably spent way more hours than I probably should have just handing around and just taking pictures of things that had nothing to do with my project, because it was so cool to see basically the cartoons that you see in your biology textbooks where you're seeing mitochondria and the nucleus and all the little ribosomes on the endoplasmic reticulum. They were there in these electron micrographs that I was seeing. So to be able to resolve that level of detail to me was just something I fell in love with. And from there, I actually had this opportunity as a grad student to then travel to Boston to spend just a short amount of time in Brad Hyman's group. He had developed a way of combining light microscopy. So kind of if you think of a traditional microscope and using light to resolve structures, with some of these electron microscopy techniques where he was physically cutting samples at 70 or 90 nanometers thick. So incredibly thin slices of tissue. And then using some traditional light microscopy techniques to resolve super tiny things that were happening. So he was actually counting individual synapses in these pieces of brain tissue that he was looking at. And so I went and learned how to do that technique in his group, spent a couple weeks, and it was just a fun experience to travel across the country and to get to learn from a different group of scientists and then take that back to my grad school project was really a lot of fun. And from there, I was hooked, and that kind of led me to applying to be a postdoctoral researcher in his group. And I've actually been in Boston now for about 10 years, first as a postdoc with Brad, and now in the last 3 years, I started my own research group, and I've been kind of slowly expanding and carving out my own area here at MGH.
Very cool. Well, it's great to get some of the detail behind your story. And I think for me, I started off with this obsession with the brain from the get-go. I'm curious how you turned towards the brain. You mentioned you started interested in molecular biology and genetics, and then maybe shifted more towards a cell biology approach when you realized that perhaps wasn't the best fit for you. What got you interested in the brain?
It's kind of funny because ASU at the time, now they've got a pretty good neuroscience department, but they really only had a couple labs that were doing neuroscience research. And I just happened to click with Stephen Hoffman. He was an immunologist and a neuroscientist who was doing some interesting research looking at systemic lupus erythematosus, so individuals who have autoimmune disease, and how different autoantibodies could affect the brain. So I just kind of had a little side project in his lab that happened to be brain focused. And I think the hooks were in at that point, and I haven't turned back since.
I think that's wonderful. And it's great to hear how you got to where you are today. And you mentioned that you launched your own independent research lab. And I know you're working on some really cool projects. So right now, Rachel, if you had to narrow it down to perhaps just tell us about one of these cool projects you're working on right now, what is the one that you were perhaps the most excited about?
Okay, so a few years ago, we were doing something completely unrelated and just imaging neurons in the brains. We had some experimental models that we were using. And the goal was just to track neurons over time. And to do that, we were looking at vasculature as kind of a fiducial marker. So we could go back to the same places and kind of reimage the same neurons over several weeks. And in doing that, we had collected just these stacks and stacks of images with blood vessels in them. And the blood vessels from the tissues that had neurofibrillary tangles, so one of the hallmark Alzheimer's disease pathologies, the blood vessels were really weird. And the normal experimental models, the blood vessels were straight, evenly distributed. They support just normal vascular functions like the delivery of oxygen and red blood cells throughout the brain. And in these experimental models that have tau, they were spiral, they're twisted, they're really irregular. They just looked weird. So this kind of observation, I think it was 5 or 6 years ago now, kind of sparked this whole research enterprise that my lab's currently undertaking, which is trying to understand what is it that tau pathology is doing to alter the structure of blood vessels, and how do those changes in the structure of blood vessels ultimately impact disease, not just the accumulation of pathology, but also the loss of neurons. So there's over 500 miles of just vasculature in the brain normally, and it's really important. If you don't have normal blood flow in your brain, it's a pretty quick time before bad things start to happen. So this hypothesis that we're exploring is just how is vascular dysfunction ultimately contributing to the neurodegeneration that we're seeing in Alzheimer's disease. And we've got all kinds of cool methods to look at this. We just did a huge assay looking at gene expression changes in blood vessels in Alzheimer's disease versus individuals who don't have Alzheimer's disease, and identified just a whole panel of genes that could be implicated in this process. And then I also work really closely with individuals at the Martinos Biomedical Imaging Center. So this is a neighboring department where they've just got some really world-class resources available to them for MRI and PET imaging. And they can do this in experimental models and in human actual patients who have early cognitive impairment. And so we're working with them to see how we can identify vascular changes in people who have early cognitive impairment. And, ultimately, I want to bring it kind of full circle. So can we develop therapeutics that could target this vascular dysfunction and use those in actual patients that we've identified that have these vascular changes that could be contributing to Alzheimer's disease? So, that, Kevin, in a nutshell, is the broad strokes of what we're working on. And I don't know if my voice is getting it across, but I'm super enthusiastic about this whole direction that our lab is taking here.
Definitely. And I really love that it was this serendipitous sort of observation of just weird blood vessels. And going back to the quote that you mentioned from your mentor, Brad, previously, going where the science takes you, this launched an entire new research area for you. I think that's remarkable.
Exactly. And this kind of interaction between this tau pathology, which is traditionally thought to be something that only impacts neurons, it accumulates within neurons, it's made almost exclusively by neurons. That link between tau and blood vessels really hadn't been made before. So it's kind of a wide open field and something that we're just super excited about.
Certainly. I think this is a wonderful area of research and, of course, particularly interesting to me because I am a neuroscientist myself as well. But to do great research, you need to have the right tools. And we'd like to take a moment to thank our sponsor, Innovative Research, and to talk about their portfolio of human biological products. Innovative Research is well known for their human whole blood, plasma, and serum, but they also supply other human biologicals and matrices, such as saliva, tears, sweat, and urine. Available donations from FDA-approved collection centers range from healthy adults to samples from individuals with specific diseases. Contact them via email at sales@innov-research.com. That's sales@innov-research.com to find out how they can provide human biologicals for your research study today. And returning to our conversation with Rachel, I think we've painted kind of a rosy picture so far of your career path and just the cool research that you get to do today. But there are down days in science as well. We can't ignore those. I think every scientist has those days where it just feels like everything's going wrong. So, Rachel, can you take us back to a time where you really struggled with something, or perhaps you had a major failure? And can you walk us through how you made it through that difficult time?
Well, let me tell you the story about our last paper that we finally were published. So it has a happy ending. This one starts probably 8 years ago. So when I started as a postdoc in Brad's lab, I mentioned we had this project where we were trying to track individual neurons over time. And I think at the time I was throwing everything against the wall to get this to work. So we had all these different compounds that we were using to label neurons. We had all these different dyes that we're using to try to label neurofibrillary tangles. Nothing was really working. In fact, I remember at one point I had tried probably like 100 different things and come up with something that kind of seemed to work. But individually, if you combined these two dyes, one that labeled neurons and one that labeled tangles, for whatever reason, they weren't compatible with each other. So independently, they were fluorescent. They did what they were supposed to do. But if you mix them together, just the signal was lost. Nothing worked, right? So it was like, okay, great. This is an impossible task. And we kind of put it on the back burner for a little while. So every now and then I would pick it back up again and try. There'd be a new dye that would come out. I'd just be continuously looking at these different biotech websites and I'd be like, oh, okay, well, maybe that one will work. And I'd give it a shot. Inevitably wouldn't work. And I'd set the project back down and then pick it back up. This went on for a while until eventually one of my colleagues, unbeknownst to me, had been trying a whole separate class of these different small molecule dyes that labeled neurofibrillary tangles and got one to work. I got one to work really well, and it was compatible with the other methods that we're using. So we were able to combine forces and use this to finally answer the problem, and did this in kind of a small experiment and got some data. So the whole premise of this project is that tau, neurofibrillary tangles, correlate really well with neuron loss in Alzheimer's disease. But there's correlation, then there's causation. So there really wasn't a lot of direct evidence to support the idea that tau is directly toxic to neurons. And it could be that tau is just something that happens to occur at the same time as other processes that are toxic to neurons. And ultimately, what we want to do is just track these individual neurons over time and see what was happening in the brain. So we finally got this experiment to work. We finally got a small data set. We submitted a paper and immediately comes back rejected.
Of course.
So this whole 6-7 year experience at this point, just immediately rejected as soon as you send it out. And that was, I have to say, kind of a big blow to me because you can't communicate in a paper to the reviewers, like just the amount of struggle this short report really communicates. So we went back, we got some good feedback. We were able to make improvements. Ultimately, what we did is we repeated the entire set of experiments again in a separate model and, made it a much better paper, sent it back, immediately rejected again.
Really?
Yes, immediately. So it's like, okay, now I'm really struggling because there's a lot of failure in science and the review process is notoriously tough. It's difficult, but it's there for a reason. And we do our best to kind of address comments. But at this point, it's really starting to drag me down just because to me, it felt like we were getting a little bit personal just because I was so invested in this project at this point. And at that point, I have a really great colleague that I work with, and he was able to take a look at this and think through some of the comments with me. And we went back to the bench, we did some more science, and we got a third data set that we were able to put into the paper. And eventually, that paper got published just recently here in the last couple of months in Cell Reports. And I'm just super proud of it. And it just represents such a struggle and just this culmination of all my years as a postdoc, essentially, in this one relatively small, but I hope impactful paper. And I think the takeaway from it, and I'd just like to share because I think it's so cool. So I told you that tangles correlate really well with neuron loss in Alzheimer's disease. But turns out if you look at which neurons are actually dying, which ones actually disappear in disease, it's not the neurons that have tau tangles. It's the neurons that don't have tau tangles. And so that's a whole other question. So what is actually killing neurons in Alzheimer's disease if it's not the tau tangles? And that's really something that we're just super curious to follow up on. And I see like 5, 10, 20 more years of research down the road here for this project.
And I think those are the best kinds of projects, right? Where you get to the end of it and it just opens the door to like million more questions rather than answering any of the questions you had.
Oh, absolutely. That's why we do this. You just want to know the end of the story ultimately.
Absolutely. I think your perseverance definitely strikes me in this story that you shared. I think there's always that one problem that you can't solve, but you also can't ignore. So what kept you going back to this problem, or why didn't you shelve it permanently and move on to something? I know in this early career stage, there's a lot of pressure to be productive, and having to keep coming back to this project must have been a challenge.
Well, to me, it just felt like it was such a central thing. So maybe it's the person that I was telling you about that didn't enjoy molecular biology. It was I have to try to understand and see physically what's going on. So to me, just this idea that we didn't have direct evidence that tau was directly killing neurons in Alzheimer's disease just felt like such a fundamental thing that we needed to know about how the disease progresses, that I felt that there was just some way that we couldn't let it down. We had to figure out a way to use the tools of science to address this question. And very lucky, again, I have to give a lot of credit to Brad Hyman because he similarly shares this fascination with trying to understand what's going on and what's happening with neurons. And so he's been incredibly supportive of pursuing and continuing to pursue this line of research.
Definitely. And I'm glad that you persevered. And of course, as you said, this story had a happy ending. And I think it's so important to celebrate these happy endings, these successes in science, because oftentimes it is a years-long process, as you described, to get to these successes. So do you have another favorite success story, whether it was a big win or just a small but particularly meaningful victory for you that you'd like to tell us about today?
A lot of what I do these days is writing based. But also some of the things that I like to do is I like to get out there and just try to troubleshoot and help out where I can in the lab. So we actually are in a very large open lab space that we share with multiple investigators. I think there's probably about 15 different PIs that lead labs in this group. And so we have a lot of shared resources. And it's wonderful because it means that we're not limited by the things that we can personally afford to purchase and have in the lab. We just have a tremendous amount of resources available to us. But at the same time, it means that everything gets used and it gets used hard. And so it breaks constantly and we're constantly fixing things. So we had this one shaker and I loved it because it was perfect. It fit my tubes perfectly and it broke. And you know what? I'm just proud of myself because I opened it up, I took it apart, I cleaned things, I fixed things, put back together, and it actually worked again. You know, it's just so nice when that happens.
You can thank your history of like playing with Legos and things in the past.
Yeah, exactly. Sometimes you just have to go in with the attitude of, you know what, I'm going to make this work and just go get it done.
We can rebuild. Yes. Well, I think these small successes can be tremendously uplifting, right? If you're having a down day, sometimes just fixing one small problem can just turn everything around.
Exactly.
Well, I appreciate you sharing these stories of challenges and successes in science. And I think you can hear your enthusiasm and you're very interested in this project. So we can tell, but I think taking a step back is important as well. And you mentioned you like to kind of get outside, get out of the lab. And I also encourage listeners to read a book to sort of expand their mind. So do you have a favorite book that you'd like to recommend for us today, Rachel, whether it's related to science or not?
I will say I love movies and books that just can transport you somewhere else. I love that feeling of just getting immersed in it and the escapist nature that these things can provide to us. So I'm going to recommend one that I just read recently that I thought was really fun, and it was the Southern Reach trilogy. The first book in the series is called Annihilation. It was adapted into a movie that had Natalie Portman in it. But the book, I have to recommend it. It'll just give you this otherworldly feeling. It features a biologist, and you could kind of sit and just churn through it in an afternoon. That's great.
Excellent recommendation. I'm excited to add this to my reading list. We'll put this on the website for listeners to find as well. And we mentioned a couple different times during your career path how you went to different places, had these opportunities to travel for science. And I think this is a major perk of the career that people may not realize is part of the career until they're already in it. So do you have a favorite place looking back, Rachel, that your science has taken you?
Yeah, conferences are great. And I would encourage anyone, given the opportunity, just take it and go because you'll get so much out of it. One that really stands out in my mind was actually kind of a smaller conference. It was the Charleston Conference on Alzheimer's disease. They did a big 10th year reunion anniversary in Hawaii, of all places. And it was beautiful. And I can't think of any other conference where I can go out and I can snorkel with sea turtles and then I can just walk across the beach into the conference room and sit and talk about science. I mean, it was really pretty ideal at the end of the day. And it was hosted by this group, New Vision Research. They're a nonprofit, and they're really just excellent, spectacular folks. And they've brought together a lot of early kind of junior investigators like myself. They really focus on collaboration and networking. And I've just gotten so much out of it. And at that particular conference, I also brought my mom. So it was really special.
Oh, I love that. So if our listeners are able to make it over to Hawaii, whether it's for science or just for fun travel, what do you recommend that they see or do?
Well, again, I'm a big beach person, so definitely go out, enjoy the water. There's beautiful fish and sea turtles swimming around. So really just enjoy taking the beauty of it all.
Oh, I love it. And is there anywhere that's on your scientific travel bucket list that you haven't been to yet, but really want to go to?
Yeah, I kind of want to go everywhere. There's these conferences and I've been in Japan recently and I've missed out on them, but I would love to go to Japan. Just thinking of places that would be a complete different experience and have the opportunity to speak with folks too that I otherwise might not necessarily be able to.
Absolutely. Well, hopefully your scientific travels will soon take you to Japan. And I think one of the things we've highlighted through our conversation is some of the amazing people that you've gotten to work with, whether it's mentors or just people that you're working with in the lab, peers, other people that are helping you in science. I think the people in science are part of what makes this career really unique and really fun. But unfortunately, there are some stereotypes about scientists. I'm sure everybody has seen them in the media, whether it's these kind of nerdy people walking around in their little white coats and kind of living in the lab. But I think a lot of these stereotypes just aren't true. I think so many remarkable, creative, funny people are scientists. So do you have a story or maybe a tradition or just an example of a memory that you've shared with colleagues that goes against some of these common stereotypes and really showcases this human side of science, Rachel?
I get rubbed the wrong way too, because a lot of the scientists that you see on TV are not necessarily like the ones that you're going to meet in the lab. We're not all kind of introverted nerds, although I might fit that description. But I have had the good fortune to work with a really diverse group of individuals, really from all nationalities. I mean, I think we've got folks from South America, from Europe, from Asia, from all walks of life. And it's been really fun to work with people who have come through and come to the lab from such diverse backgrounds. And this has made for some really fun lab traditions. So over at the Martino Center, one of the things that they do for Thanksgiving is share recipes. So they'll come out with like a recipe book every year. And it's really fun just to see what a Thanksgiving dish would be if you come from a completely different country that doesn't necessarily celebrate Thanksgiving. So I think that's been a fun tradition, is just kind of enjoying each other's kind of cooking and things like that.
I love it. So have you experimented with some of these recipes?
To a certain extent, yes. Yeah.
I'm not a good cook, so I marvel at people who are able to adapt recipes and do really cool things in the kitchen.
I think my problem is I'm not a particularly good cook myself. So I think a lot of the spirit of the recipe gets lost when I make it.
It's all down to the measurements.
But I can appreciate it when other people make their favorite dish. So.
Well, it sounds like you've got some really great traditions there. I love this idea of collecting things from all these different cultures and these people who come through the lab in the center and really putting together this showcase almost of the different cultures and the different people who have been there over the years. So I think that's amazing. And I know in science, you're answering some really remarkable questions we've talked about earlier in our interview, but there's often barriers, and we've touched on some of them today, whether it's just time, funding, technology, not having the dyes you need, whatever the case may be. So Rachel, if we took away the barriers that normally hold you back, what is the one question or the one problem that you would most want to solve?
Yeah, I mean, whether it's something related to vasculature or not, I really want to figure out why neurons are dying in Alzheimer's disease. I mean, there are these cells that are incredibly long-lived. You're born with them. Why in the later stages of life do they suddenly start to be lost? You know, I think there's a lot of technologies that are really coming to the forefront. So things like spatial transcriptomics and the ability to look at proteins in individual cells with such a level of detail that we couldn't even just a couple of years ago, that I'm really hopeful that this is something that we can start to nail down in the next few years here.
Absolutely. I think it's been really exciting to see advances in the field of neuroscience as a whole, but particularly in the field of Alzheimer's research. So I think this is an area that a lot of people have their eyes on at the moment.
Yeah, definitely.
And we've covered a lot of ground and talked about a lot of different aspects of your career. We'd love to end by talking about advice. So for our listeners out there who are perhaps on their own scientific journeys or just their own journey in another career in life, what is a piece of advice, Rachel, that you received at some point that really helped you that you can share with all of them today?
Yeah, whether or not it's just you're trying to figure out where you want to go, what your next job is going to be, or who you're going to hire on your team. So I'm in a position now where I'm looking to expand the lab and form my own research group. Something that's really been impressed upon me is just the importance of finding the right people and forming that team. And I'm a big supporter of teamwork. I love working as a group. I don't think I would be successful if I hadn't reached out to people who are interested in a lot of the same questions and gotten their input on some of these and gotten their help with a lot of the projects that have been successful. And so just really finding the right people. And those people might not necessarily be the ones who on paper look the best, myself included. It's always amazing to me that I have to look back on my career and just think about how I started out in this relatively small program at Arizona State University. And I've just kind of steadily moved up to a place where I have a tremendous number of resources, and I've been incredibly fortunate here. But at no point was I really feel like I was really necessarily the strongest candidate on paper. So I have to thank the people who took a chance on me and just reflect on that and think about how moving forward to form the best team, because the best team is always going to get you the best result and do the best work.
Oh, absolutely. So just to dig into this a little bit deeper, Rachel, how do you find the right people? I think it's easy to say, like, you've got to build a good team, but what do you look for maybe when you're looking at these applications? And like you said, perhaps the person who is not the star candidate on paper is the one that would really make the most impact in your lab. How do you kind of make that connection and find those people that you're looking for?
I don't have a magic recipe for this. So my disclaimer is I still haven't quite figured it out myself, but I have had the best success when I just spend the time talking to folks and asking a lot of questions and really listening to them. So going to conferences and speaking to individuals, that's actually where a lot of the people from my lab have ultimately joined from, is just folks that I met at conferences, and we stayed in touch, and it ended up being a really great fit ultimately.
I think that's wonderful. I think meeting people, making those connections and networking is something that people don't always emphasize so much in science, sort of as a career development mechanism, but I think it is tremendously important.
Yeah, definitely. You have to get to know the folks that you're going to spend so much time with. So it can be tough to know beforehand if it's going to be a good fit or not, but just really taking the time to speak to individuals and not just kind of glance at a CV and make a snap decision, I think is really important.
Absolutely. Well, excellent advice for listeners out there. Is there any other last piece of advice or perhaps a last note of inspiration that you'd like to leave everybody with at the end of our conversation today, Rachel?
If you feel like you're failing, you're probably doing science right. I mean, I fail every day. So just keep at it, just persist, and it's eventually all going to work out. So you're not alone.
Well, I think that is a wonderful note to end on. So true in science. And if our listeners want to learn more about you, your lab, and the wonderful work that you're doing, what is the best way for them to do so?
You can find me on LinkedIn. I'm pretty easy to get in touch with that way. And I also have a website. It's Bennett Alzlab, ALZ as in Alzheimer's.com.
Excellent. Well, listeners, definitely check out Rachel's website, learn more about her research, and get connected on LinkedIn. And Rachel, it's been such a pleasure to have you on the show today. Thank you so much for joining us.
And thank you.
I'd also like to take a moment to thank all of our listeners out there for tuning in. I hope to connect with you again in another episode of People Behind the Science.